Good morning, everyone. First of all, let me thank for selecting me for this very prestigious prize. I'm deeply honored. I dedicate this prize to my research group. We are a relatively young research group. My group was established in 2015 at the University of Debrecen, and this award indicates us we are on the right track, and we would like to continue our work at an even higher level, if it's possible. I would like to indicate that I'm not yet part of the mentorship program, so if you would accept my application, I would be happy to be part of this community for the future. Today, I was instructed to give you a very specialized talk, which is related to our recent publication shown here. And I have a dilemma, because this is an overly specialized topic published in this journal recently. However, the audience is a non-specialist audience. I see many young faces, and I'm here to encourage you to be involved in science, not to distract you from science. So if you look at the panel on the right-hand side, this is a word cloud panel. Everyone can try at home. You just copy and paste whatever text you have to this program, and the frequency of the words is proportional to the height and the height of these words. And I just copied the text of our paper, and you see there are many special terms you are probably not familiar with. So heterochromatin, the NDX protein, I don't know who knows what these are, or methylation, DNA methylation, or chromatin, high-C technology, chromosomal contact. So it's extremely specialized topic, and how to explain this in 20 minutes. So I decided to step back, and I would like to try to start with explaining the basic question, the big question. So we are studying the structure of chromosome, the DNA. The DNA is an extremely long polymer molecule. It's like height of a man or a woman, like this very long. And this is compacted to a very tiny volume, the cell nucleus. So in this first simulation, in this performance, the contortionist lady represents a chromosome, okay? The box here represents the nucleus, which contains the chromosome, and obviously the chromosome must fold up to squeeze into this tiny volume. Actually, the box is at least 1 million times higher than the volume of the cell nucleus. Okay, so here we go. You see there are some intrachromosomal contacts, so the legs are in contact with the shoulders now, and I speed up a little bit, so I tell you in advance, so the lady squeezes herself into the box. But the situation is even more complicated when we have a chromosome in the box and there's another chromosome coming inside. So we need to take into consideration the interchromosomal contact. So the different chromosomes make contact points with each other, and during this process, the function of the genome must work. Okay, so the transcription apparatus, DNA replication processes, DNA repair processes, epigenetic processes must be functional during this process. And it's almost done. A little bit more, yeah, okay. Okay. And there we go. So the chromosome is compacted in the nucleus. And now I show you some real data. We established for the first time in Hungary a special genomic technology. It's called genome conformation capture, or chromosome conformation capture technology, HiC, in which we can map contact points between any chromosomes. And from this three-dimensional map, we can reconstruct the 3D structure of each chromosome. So this is chromosome X in mouse, and these balls represent some structures that are mapped onto this 3D structure. So in very short, in this cartoon, the red structures represent, for example, proteins that are involved in making these contacts. The blue structures represent other proteins that are not involved at all. In this cartoon, the red structures establish interchromosomal contacts, but not the blue ones. And our interest is to find some of these structures shown on the right-hand side cartoon. So we are interested to understand the role of histone-modifying enzymes and histone modifications that establish these contacts, and special nucleic acid structures called R-loops. This will be the focus of the current talk. So the R-loop, which is a three-stranded nucleic acid structure, which is composed of an RNA-DNA hybrid and a displaced single-stranded DNA. And also we are interested to map and understand the formation of double-stranded DNA breaks. And as shown by this cartoon, these three processes are interrelated with each other. So we want to understand causal relationship between these two processes. Histone modification can induce R-loops, which can lead to DNA breaks, and vice versa. We can start from any point, basically. So there is a complex relationship we would like to understand. But the current talk, as I mentioned, is focused to the R-loop field. So what is the R-loop? So this is the structure, the prototypical structure, what I showed in the previous slide. And we would like to understand where these structures form in the genome, why they form there and not elsewhere, how they are recognized by certain reader proteins, how the R-loop signal is transmitted to induce chromatin signaling processes, and finally, how the formation of this structure leads to human disease. So these are the big questions what we are interested in. And from the literature, we learned that when these R-loop structures form on the wrong place and wrong time, they can lead to human disease. So in this side of the coin, you see mutagenesis, DNA breaks, hyper-recombination, genome instability, many bad stuff, basically, which are or might be related to human degenerative diseases and cancer. On the other side of the coin, we learned from recent literature that they are required for transcription, repair, replication, and even chromosome structure, which is our main focus. And to understand the biological role of these R-loop structures, we need to find factors that positively or negatively regulate the formation of these structures. And this is what our paper is about. And we use different approaches from powerful yeast genetics to model organisms, human cell lines, and even patient samples from biobanks. A good way to find R-loop regulatory proteins is the old-fashioned way. You don't need so much, you need to read scientific papers. It can be still very efficient. And basically, in 2015, when my lab was established, I came across this very nice paper published in in Science. It came out from the Caroline Dean's lab, and for the first time ever, they found an R-loop stabilizing protein called Nodulin homeobox. At that time, there was no protein. We didn't know any protein that regulate R-loops. It was before the screens and mass spectrometry data that are coming out nowadays. It was the first ever established R-loop regulatory protein, the Nodulin homeobox protein. And I thought, oh, it's a very nice paper, and I happened to know Tibor Csorba, from Gödöllő, who was a postdoc at the Caroline Dean's lab at that time. And I said, Tibor, okay, why don't we team up? I don't know anything about this model organism, which is Arabidopsis. I don't know nothing about this, but you are expert on Arabidopsis genetics. You have access to all of the mutants, all of the biological material, and we can do genomics and molecular biology. So let's team up. And we decided to understand the genomic role of the NDX protein, not only in the case of this specific gene, because this paper was focusing on a single gene, the flowering locus C gene, which regulates flowering time in Arabidopsis. And the basic finding was that the activity of this FLC gene is regulated by a non-coding RNA coming from the different direction, but the promoter of this non-coding RNA forms an R loop, which is stabilized by this NDX protein. So NDX negatively impacts on the activity of this gene. And I thought it is remarkable because if NDX can stabilize R loops on the FLC gene, probably this is also true at the genome-wide level. And probably if we can clone the NDX gene and humanize it, we can make a tool to target human R loops, and maybe it can be good to on the long run to cure human disease related to R loop formation. So we had very big plans with the NDX protein, and we started our project in 2015. And in the next slide, I show you how this project was after 4 years of workload. Okay, so this painting just represents that it didn't went good. Okay, so in 2019. We need to reconsider everything. Whatever experiment we did and whatever papers we submitted, it was rejected because it turned out we were absolutely wrong about the role of this NDX protein. The starting point was wrong, and we realized this mistake after 4 years. Why? Because it turned out that the NDX protein is not a general R loop binder protein as we supposed. It has a very specific role in the case of a few genes only. But at the genomic scale, it has nothing to do basically with the R loop structures. So in this cartoon on the Venn diagram, I show you the overlaps between the R loops in red on the genome and the NDX protein. Shown in green, and you see that the overlap ratio is rather small. Most of the binding site of the R loops and the NDX protein do not colocalize at all. On the right-hand side, you see a genome browser cartoon. So these represent the physical map of the genes. The green signal is the enrichment of the NDX protein over the chromosome. So you see some peaks here. The red one is the R loop map, and there is little overlap between the two. So it was kind of shocking experience for us. So if it's not the regulation of R loops, then what is the function of the Nodulin homeobox protein? So this was the big question. When we annotated these binding sites, which means that we determined their enrichment in certain genic regions, shown on the left-hand side cartoon. So each row is an annotation category, gene promoter, 5' UTR, and so on and so on. So we observed that there is an antagonism between the annotation of the R loop structures and the NDX protein. Namely, the R loops are enriched over the gene promoters and 3' UTRs, so basically these gene regulatory regions. So this is the promoter, which is R loop rich, and the 5' UTR is part of the first exon. So most R loops were associated with these regions. However, the Nodulin homeobox protein was associated with non-coding regions. So several parts of the genome is a non-protein coding part. these are structural parts involving the centromeric regions. So the NDX signal was very enriched over the pericentromeric regions. The centromeres are involved in chromosome segregation. And they are highly repetitive. There are very few genes along these regions, but they are enriched with transposons. And we also found that the NDX protein was localized mainly on these pericentromeric transposons, which are basically jumping genes. They are not protein coding genes, they are jumping genes, they are parasites that are embedded in the genome. When we computationally reconstituted the functional chromatin state of Arabidopsis based on these biochemical features like histone modifications, the chemical modification of the DNA, GC methylation, also the transcription output, histone variant. So all of these features were combined by a mathematical methodology, and we established 9 functional chromatin states, from which the first 2 represent the mostly active chromatin state, so-called euchromatin, okay? And the last 2, 8, 9, represent the inactive chromatin, which is the constitutive heterochromatin. I hope you appreciate that the R-loop structures are mainly enriched over the euchromatic regions, so the open regions. However, the NDX protein is mainly enriched in the closed rigid chromatin region, which is typically the constitutive euchromatin. So again, we see an antagonism between the location of R-loops and the NDX protein. At higher magnification, if you just look at the chromosomal spectrum, just look at the green spectrum, which is the enrichment of the NDX protein. And below, you can see the distribution of the so-called heterochromatic small RNAs. These are small RNAs that regulate the activity of transposons. They have nothing to do with proteins. In a process which I will not elaborate today, please just remember that these sRNA loci that regulate transposons have different types, and the green signal, which is the NDX, was mostly colocalized with these guys here, so sRNA 6 to 9 category, which are the typically so-called heterochromatic small RNAs. So this one here, that regulate the activity of these parasite element, the transposons. And again, the R-loop signal was overlapping significantly with the first 2 small RNA category, which has nothing to do with the regulation of transposon activity. In the next stage, we decided to do some functional assay. So what does it mean in research? We took the NDX gene and we destroyed it, okay? So we inserted this construct into the gene. It's called insertional mutagenesis, and this gene was no longer inactive, and we compared the mutant line with the control line, okay? So we had a mutant line indicated in red in the below cartoon, and the control sample is shown in black. And we performed so-called small RNA sequencing, which showed us the activity of these small RNA loci that I showed you in the previous cartoon. And I hope you can see that in terms of the length of this RNA, or in terms of the functional groups of this RNA, they are mostly upregulated in the NDX mutant. It's also visible in this clustering cartoon. So most of these small RNA loci that regulate the activity of these self-parasite transposon elements are upregulated or induced when the NDX gene is inactivated. It means that the NDX has a role in the suppression of these elements. So what is the epigenetic basis of this phenomena? maybe you heard that methylation of the cytosine basis on the DNA is a well-established epigenetic signal. And when this methyl tag is there, it means that the gene is shut down. So the methylated cytosine is antagonistic with the activity of genes. And we did so-called BS sequencing experiment in the control line and in the NDX mutant line. And in these circus plot figures, first of all, the perimeter of the figures are the concatenated chromosomes. And you can observe blue dots and red dots. And the blue dots along the circle represent the genomic regions that are hypomethylated in the NDX mutant, whereas the red dots represent hypermethylated DNA regions. So I think it is very obvious that in the NDX mutant compared to the wild type control strain, we have methylation changes, and we found these hypo-DMRs, hypo-differential methylated regions very interesting. Why? Because they perfectly co-localize with the binding site of the NDX protein. So these enrichments for the hypo-DMRs perfectly co-localize with the NDX protein. Therefore, probably it is the direct function of the NDX protein to regulate the methylation state of these sites. I said that we are interested in 3D chromosome architecture, so we asked what is the structural basis of these molecular changes, and we performed this genome conformation capture experiment in the wild type control strain, in the NDX mutant strain. On the right-hand side cartoon, you see the ratio of these two maps, which is by visual inspection is no different, but actually they are very different. And here, the red color indicates dominating 3D contacts in the NDX mutant, which is not present in the wild type strain. The blue is the opposite. And we observed that there are interchromosomal contact changes in the NDX mutant, represented by this knot model. So there are some newly established de novo interchromosomal contacts. And we know that this knot structure is a site in the nucleus that inactivates transposons. And we not only observed de novo knot interactions in the NDX mutant, we also observed that these interactions are quite similar to interactions that were observed in DNA methylase mutant. For example, the DDM12 mutant or the MET1 mutant, which are well-established mutants that inactivate the DNA methylation pathway, show somewhat similar high-C pattern as our NDX mutants shown here. For example, if you look at these positions, it's empty in the control strain, but you see these dots represent de novo contacts in these DNA methylase mutants and also in the NDX mutant. So the final conclusion of this paper, which was recently published, was that NDX is a master regulator of the heterochromatin in Arabidopsis, and its function is coupled, it's functional coupled to the DNA methylase pathways and the heterochromatic small RNA expression. So briefly, that was the finding. I haven't shown data on phenotype. This is still a project which is ongoing. We see some very weird phenotype in the NDX mutant, which is transgenerational. So there are some very remarkable changes. The flower structure is completely changed, and we would like to understand why this is happening. But as suggested by these results, it is epigenetically regulated, which occurs through DNA methylation. I don't know how much time I have, so it's over. I would like to thank to my collaborators, and this is my group, and thank you very much for your attention.